is right to the sea. Come on in and don't be shy. Just to make your day complete. You might get baked into a pie on the magic scuba. Step inside, it's a wild ride. Come on, ride on the magic scuba.
The excretory system is necessary to maintain homeostasis because it helps to remove cellular waste and helps to maintain salt-water balance. This is necessary because it gets rid of urea, carbon dioxide, excess salt, and excess water that can be dangerous to the body if left there, so it helps to maintain a stable body condition. These compounds are filtered from the blood by the kidneys, which combine them to form urine. Evolution of the kidneys give an overview of evolution of the excretory system as it represents an important step in the evolution of invertebrates to vertebrates. Paleontological evidence suggests that the earliest vertebrates survived in fresh water, causing them to inevitably absorb excess water by absorbing it through skin or swallowing it with food, so a mechanism for eliminating the excess water was necessary. Also in this fresh water, salt was scarce, so it was necessary to prevent wasting the salt in the body. The elimination, water, and reclamation of salt may have been the earliest functions of vertebrate kidneys. These were tufts of blood vessels filtered water out of the bloodstream into the body cavity and convoluted tubules with openings into the column and collected the filtrate and retrieved any salts from it. These tubules arrived as localized modifications of blood vessels, eventually leading to the capillary beds that surround the kidney tubules.
Each of the ways of excreting nitrogenous waste comes with its advantages and disadvantages. The simplest form of excreting waste is by excreting ammonia. By excreting ammonia, the organisms excrete one nitrogen atom per molecule. Ammonia, however, is highly toxic to living organisms, which is why it must be flushed with a lot of water. Because of this great demand of water, excreting ammonia is most commonly found in aquatic organisms as they are surrounded by water which they use to flush out the toxin. This method is the most energy efficient as it requires almost no energy to transform the ammonia. While this method is best at saving energy, it is the most toxic, therefore requiring a lot of fluids. Another method that organisms excrete nitrogenous waste is with excreting urea. Urea is more efficient at transporting nitrogen as it contains 2 nitrogen atoms per molecule, as opposed to 1. Along with being more efficient, urea is also very water-soluble and 100,000 times less toxic than ammonia, so it requires significantly less water to be eliminated. However, it costs energy to form the urea and it is still slightly toxic, possibly resulting in harming organisms if their kidneys are too weak, as it would cause a urea buildup in the blood. Because it requires less water, land animals can use this method as they do not need to be flushing out highly toxic ammonia. The least toxic form of nitrogenous waste is uric acid. Uric acid has 4 nitrogen atoms per molecule and is non-toxic. However, it requires the most amount of energy to create due to it being a very complex metabolic process. Because uric acid is non-toxic and non-soluble, it requires a minimal amount of water to be excreted out of the body, but because it is non-soluble, it can build up and crystallize in organisms, causing complications.
All organisms achieve osmoregulation by regulating salt and water balance within their cells and tissues. Before we talk about osmoregulation, we have to remember that osmosis is the diffusion of water across a semi-permeable membrane. Remember that it is vital that our cells remain in an isotonic environment where the concentration inside and outside the cell remains the same. When cells are in a hypertonic environment, the water will rush out of the cell, causing it to shrink or shrivel up. Conversely, in a hypotonic environment, water will rush into the cell, causing it to burst. In both instances, the cell is damaged. However, when the cell is existing in an isotonic environment, there is equilibrium, and the cell is happy. Now let's talk about osmoregulation. Osmoregulation is the active regulation of the osmotic pressure of an organism's fluids to keep an organism's water content constant. This simply means that it keeps an organism's fluids from becoming too diluted or too concentrated. Humans are osmoregulators. This means that they firmly regulate their body osmolarity, which always stays constant. Osmoregulators actively control salt concentrations regardless of the salt concentrations in the environment. Fish are also osmoregulators. However, we see differences in the osmoregulation of freshwater and marine fish. For fish that live in freshwater environments, the saltier area is inside the fish. As a result, they are going to keep having water flow into them. Freshwater fish have urine that is very diluted. Saltwater fish, on the other hand, experience the opposite problem. Here, there is a higher concentration of salt outside the fish, so water continuously flows out of the fish. As a result, saltwater fish have to actually drink the salt water. Their urine is very concentrated. Sharks and rays are something else. They are osmoconformers. The osmolarity of an osmoconformer is the same inside the organism as it is in its environment. They do not have to regulate osmolarity. As a benefit, they do not have to use as much energy as osmoregulators in order to regulate water content. However, a disadvantage is that they are subject to changes in the osmolarity of their environment.
Bill Nye the Science Guy.